Starting out very basic, was, trying to figure out how to format this presentation. Then I realized if we don't at least cover the most basic things at first, which I was planning to cover at the in-person trainings, it would just seem a little out of place. It would seem like we were skipping something. So very quickly, I'll just talk about what a lichen is. And lichens are symbiotic organisms composed of multiple separate species. So you have always in a lichen, you at least have a fungus, and you have what is known as the photobiont, and that is either an alga or a cyanobacterium or both. So that means you have something that photosynthesizes. And then they found fairly recently, as in within the last 5 or so years, that a lot of lichens also contain a third partner, and that's a yeast. So yeast are also a fungus, but a completely different type of fungus than the one that is kind of the centerpiece of the lichens. So lichens are they've kind of gone back and forth from being considered in the botany component of science to the mycology component of science, and really they better fit in with mycology, which is the study of mushrooms, because it is the mushroom that determines the species. So a lot of lichens actually share the algal or cyanobacterial species that is within them, but each different lichen has a unique fungal species. So it's the fungus that really determines the species, and that's fairly key. What's really cool about this symbiosis is that it allows for lichens to exploit a variety of niches and to persist under really extreme conditions. So you wouldn't see either algae or fungi growing on a rock, for example. Neither could persist in that environment. Of course, a fungus can't get any nutrients from a rock. We never see fungi growing in rocks or growing on rocks. And algae, while they might grow on rocks for a bit and photosynthesize, they would die off fairly quickly. So together they can persist there for decades or possibly even centuries. And this is a rock that I took a photo of at a brief layover in Phoenix a couple weeks back. And so this is in the Sonoran Desert, right under a saguaro. So really extreme environment, hot temperatures, really dry. But these are lichens. This is about 5 different species that you can see here on this one rock, persisting in a desert environment. So they're pretty incredible. And what allows them to persist in these places is the ability to essentially fully desiccate. So without any rain or any water, they can just stop growing. They shut down all growth, they close up, and it's the fungus, which really makes up the exterior of the lichen, that allows them to do that. So by doing that, they can persist even in the vacuum of space. So lichens have been taken up into space and exposed to space on the outside of a spaceship, I think on the International Space Station, and they can persist out there. And then once they're brought back in, rehydrated, they're still alive. So they can really deal with the incredible radiation that can be found in outer space. So pretty crazy. But what's also really interesting about that is, on the other hand, they're exceedingly fragile. And that's part of what this project is exploring and looking at. Oh. So why lichens for this project? I've been wanting to do, and I think I mentioned this, a community science project for a while out at Mount Pisgah. And one of the things that was really important to me was to do something that would engage the public in both learning and then also in contributing something that could be, actually valuable science. And so as I began to look more at lichens, realized that we have a really impressive abundance and diversity of lichen species on Mount Pisgah. So well over 100 species. Not all of them, as I mentioned before, are readily identifiable. A lot of them require chemical analysis or microscopy to separate. But we have a really high diversity. lichen communities in the Willamette Valley are heavily fragmented by both land use change and by atmospheric pollution. So this is where we really get into the component of lichens being actually quite sensitive. So when they're desiccated, when they're dried out, they're essentially in kind of a hibernation mode. They're not doing any transfer of gases with the environment. So no gases in, no gases out, really. So totally good in space, totally good in desert environments. But when they are getting moisture and they are growing, then they're exchanging gases consistently with the environment. And that's really how they're able to survive, because they don't have any roots. They're not extracting any nutrients from the soil. Everything is coming from the atmosphere, from the sun, and from micronutrient deposition and rain. So the problem there is if you have really high amounts of air pollution, some lichens are extremely sensitive to that. We have some lichen species that absolutely love it, and those are the ones that you tend to see in urban environments. If you're in Eugene and not on the outskirts, you have fairly low lichen diversity, and you're going to see a lot of species that are kind of bright yellow or orange. And those tend to be the ones that really love nitrogen. So they love high amounts of nitrogen. The rest of lichens, the ones that are not nitrophilic, really have a difficult time with nitrogen for the most part. Some are able to handle it to a degree. Others really can handle almost none of it. So they have been used as bioindicators for air pollution, and a really large amount of pioneering work has been done on that, a lot of it coming from Oregon, a lot of it coming out of the Pacific Northwest. And they've actually found ways to pretty precisely use specific lichen species as bioindicators for air pollution. And I think just really quickly, oops, I keep doing that. I think I'm going to back out of this and share a different screen to show you what that looks like. 1 second. So only allow me to share my desktop. Okay. Can you see, oops. Can you see this now?
Yes.
Different website. Okay, great. So this is a website from the Forest Service that is looking specifically at different lichen species and their tolerance to atmospheric nitrogen. And basically graphing that here. So we have this lung lichen, the really common one that you see all over Mount Pisgah, Loberia pulmonaria. And it has a low to moderate nitrogen requirement, with peak detection occurring around 2.2 kilograms of nitrogen per hectare per year. So that's the basic metric that's used there. But you get a lot that have their peak detection essentially at the lowest level of nitrogen deposition per year, and that's about 1.1 kilograms per hectare. And so you can use this to get an understanding of the air quality in an area. And then you can also, just by that understanding that a lot of these lichen species have a really strict requirement of air quality, you can begin to recognize which ones are potentially at risk in an area like Mount Pisgah, that's on the edge of the Willamette Valley, really highly intact lichen community, but also really close to an urban center that is growing, and where air pollution is a real factor. And of course, this is not the kind of air pollution, I was born in Riverside, California, outside of LA, not the kind of air pollution at all that you see in places like that, where you have this dense smog, right? It's less detectable, less noticeable, but you can really detect it through these lichen communities. So part of what this project is looking at is what lichens that are fairly sensitive are still persisting right now. And so those are the ones that will be targeted. So I'm going to go back to sharing just the PowerPoint here for a moment. So they're great bioindicators. And then the other thing is that the lichen flora on Mount Pisgah is not well documented. We have really good documentation of the plant species. We're still finding some new ones that we hadn't found yet. But people have been really documenting the flora on Mount Pisgah for 50 years now. The mammals are well known, the reptiles are well known, amphibians are well known, the birds are well known. But the lichen flora is really still fairly not super well documented. So that's another nice reason for this project, just to get kind of an understanding of what's there and where different species are distributed. And then the other reason that lichens are great for a project like this is that identification is fairly accessible with a little bit of training. There aren't that many different things to look at in a lichen to kind of identify it, at least the ones that don't require chemical analysis or microscopic analysis. The other reason is that they're really cool. So lichens are just neat. They have a bunch of different forms, and this one is one that, to my knowledge, we don't have on Mount Pisgah. You tend to see it in wetter conifer forests, but this one is the common name that I really enjoy is fairy barf. And that's, more or less what it looks like. It's just this kind of spray of lichen across often logs, or in this example, it's on top of moss. But they're very, very odd organisms, and they have persisted for hundreds of millions of years and diversified into about 20,000 species worldwide. So really hugely diverse. So what you'll be doing is photographing lichens along trails at Mount Pisgah. We'll talk a little bit more about what along trails means, but the idea basically is not to go too far off trail, to essentially look at things that are trail side. I'll also ask you to document the tree species when possible. So we'll go over a little bit of tree identification as well. That's really not a requirement. That's sort of, if you're confident about the tree species that a lichen is growing on, then that's awesome to include. Part of the reason for that is that one of the big threats to lichens in our region in the next decades is the emerald ash borer. So the emerald ash borer is an invasive beetle that first showed up on the east coast of North America in the late 1990s, actually kind of around the Detroit area, and then spread rapidly across the east coast. And in its native range in Asia, it has evolved with the ash trees there, and the ash trees there have resistance to it. However, our ash trees in North America have no resistance to it, and it's almost complete. It's an extinction level event where it removes 99% of ash trees of all North American species when it gets into an area. And the emerald ash borer was just found in Forest Grove, Oregon, which is the first record on the West Coast. It's been there at least a few years now and has likely spread a bit already. Their spread is going to be spotty. It doesn't all happen at once. And there are plans for contending with it and reforesting areas with hopefully resistant ash seedlings eventually. But a lot of our lichen communities in the Willamette Valley, a lot of the diversity can be found on Oregon ash trees. So partly a component of this will be recording the tree data, and it's particularly interesting if it's on Oregon ash trees. Then the other thing is uploading observations to iNaturalist, which we'll be covering. And that's kind of really what I want to talk about tonight. It's a lot easier to show how to do that on a computer screen, since iNaturalist runs through either a computer or through your phone. And then you have the option to make identifications to the degree desired. So you don't have to, and I don't want you to be held back by not being able to identify lichens. These trainings are to give you the tools to be able to do so, to start learning. But it's not a requirement of the project to be able to identify lichens, and I don't want you to feel frustrated if you can't. Some of them are fairly challenging. And so what I'll be doing in this project primarily is identifying your observations on iNaturalist. So I'll be looking through all of those. There will be other people who will be helping out with that as well. I'll provide training and resources, and then provide feedback. And specifically, you know, if you have questions for me, I want to make sure that I'm available to answer your questions, to help you out with any additional kind of training you might want. And then I'll also be asking for your feedback. I'm also going to be tracking the volunteer effort. So I'll make sure to count your hours. And what I'm thinking is that basically whatever time you spend out looking for lichens, photographing lichens, and then making those iNaturalist observations, just send me an email when you do that. That can be once a week, or if you're going out, you know, once every 2 weeks or once every 3 weeks, whenever you put in, you know, an appreciable amount of time. And I'll send reminder emails to check in about that. But, you know, a component of this is we want to track the volunteer effort there too. And then all at the end, provide a summary report of what's been found. Whoops, went too far. So question from Yona. The project on iNaturalist, not sure of the name of the project. I will show that in just a moment. And please send me an email too, and then I can send you a direct link to it. And so I'll talk about that a little bit more in just a second. So the trainings, the first one's tomorrow, and the next one is on Saturday. We'll cover basic lichen identification, basic tree identification. We'll look at how to photograph lichens, and then how to look for lichens. And so that is actually, I think, one of the more important skills is just kind of to know where to look to find some of these sensitive lichens. We'll be looking predominantly at lichens that have the cyanobacterial partner. These are ones that fix atmospheric nitrogen. They are less common in the environment usually, and they're a lot more sensitive to air pollution, almost all of them. But then there's some other lichens that we'll be looking at as well that aren't cyanobacterial, but have shown themselves to be really sensitive to air pollution, and they're good indicators of essentially good lichen habitat. So we'll be going through all of that, and I will give you tomorrow a hit list of lichens to target, and that's 12 lichen species that I've made in a little laminated brochure. These laminated brochures are homemade, so they are imperfect. I don't fold things well, but they're good enough. It's also really hard to fold laminated paper. The idea behind the lamination is that you might be going out in the rain too.
Question.
So, yes.
Question. So is tomorrow going to be all indoors, or should we be prepared to go out in the rain?
Be prepared to go out in the rain. Yeah,
Okay.
it's going to be mostly outside. That's the plan. So if it's really dumping tomorrow, we will potentially find a way to spend a little time inside, but the idea is to mostly be outside.
Thank you.
So for Friday and Saturday, be prepared for some rain. Yeah. It looks like the weather forecast is decent, but it's been switching back and forth over the last couple of days. So briefly, I want to mention that the Howard Buford Recreation Area, which is also known as Mount Pisgah, is a component of a few different land management agencies, essentially. So the Mount Pisgah Arboretum, which is this orange highlighted area here, and that's where I work, is a 209-acre kind of nature education facility. And so we manage this area. The entire area is owned by Lane County Parks. And then the Friends of Buford Park, which is a partner organization, contracts with Lane County to do stewardship projects and trail work and other things throughout the Howard Buford Recreation Area. Also has a fantastic native plant nursery and all of that. So it's a big mix of organizations and land management. And then also, some of you may know this, but you can see the border of Howard Buford Recreation Area here, and especially on the backside, it doesn't actually cover the entirety of the hill that we call Mount Pisgah. This backside here out to the confluence of the Coast Fork of the Willamette and the Middle Fork of the Willamette River is currently owned by the Nature Conservancy, and that was purchased back, I think, in 2008 or 2009. And that should be transitioning ownership fairly soon. I think everyone knows enough that I can say it, but probably to Mackenzie River Trust. But it's a really long process of transitioning ownership. And this is another 1,400 acres, basically. So huge amount of property in public ownership. Why I'm showing this map and what I've highlighted on this map are places that I think would be good to look for some of these lichen species, these target lichen species. So I will send this map out in an email, so you don't have to squint to look really closely at this right now. But I just wanted to show this kind of in a broad scale to give you an idea of some of the places to look and why. And primarily, so if we look, you can see my cursor right here in the Arboretum, right around this 35. This is a really fantastic spot for some of these sensitive lichen species. And part of the reason for that is it's a north-facing slope with a creek drainage, and the air from the river just moves up this creek drainage. So it stays humid for much longer throughout the year, always maintains a higher humidity than some of the surrounding areas. And if you've ever, I imagine a lot of you have walked the summit trail up to the top of the summit. And if you know this kind of where the trail curves, it makes a, right after that really steep section, it makes a hard curve to the right. That whole area there where you're kind of looking down appears to be a really old landslide. And so the water from the river kind of gets sucked up that drainage where there's now creeks running down that old landslide. And just that slope really moves that air up that drainage. So you get some really interesting lichen communities here. And using these ideas of these microclimates where you get a lot of interesting lichen communities, you can extrapolate and kind of find where you might see similar communities. And so we really want to look, and I'll zoom in on one of these, at places where, whoops, gone too far, at places where you have kind of a north-facing slope and creek drainages. And of course, all of our creeks are seasonal on Mount Pisgah. We don't have a single creek that runs year-round. So these are really just drainages of really kind of excess runoff when the water from the rain hits the bedrock. But they create some interesting microclimates. And particularly the trees in our area that host most of the sensitive lichen species, and usually the highest diversity, are hardwoods. And that's not true at all if you go into the along the Oregon coast or up into the Cascades. Of course, the tree species composition totally changes there, but you tend to see conifers hosting some of the same species that we have here in the valley that tend to prefer hardwoods. But for whatever reason, in the valley, they're on hardwoods. And so a lot of the areas that I've highlighted, pretty much entirely, are areas that are tend to have good hardwood forests, or at least mixed forests. So now we'll talk about, we'll talk about using iNaturalist really quickly. And so just a quick overview of iNaturalist before I actually show you what that looks like. iNaturalist is a nonprofit initiative of the California Academy of Sciences and the National Geographic Society. And it actually started as a graduate project from a few graduate students in, I don't know, I think around 2013 or something like that, or maybe even a little earlier than that. And it's a platform that's based around essentially crowdsourcing biodiversity data from observation to the identification. So anybody who logs onto the website or downloads the app can submit an observation of any plant, animal, fungus, whatever, and have the opportunity for it to be identified on the site. And anybody can serve as an identifier on the website too. So it's not all experts that are identifying. It's crowdsourced identification and identification by consensus. What's neat about that is all of this biodiversity data is transparent and it's open source. So you can see the route to identification, particularly on ones where there's kind of disagreement initially over identification. And you can see that discussion, which I think is really neat and really instructive, particularly for people who are just learning. And it's great too, because it also shows that people who are experts in their field make mistakes. I think that's really important. Of course, some people, you do have the option to delete your identification or comment. But I think the spirit of the website is to generally maintain that. And I think it's nice to have a record of mistakes and the route to get to a hopefully correct answer. It's an awesome tool for learning. And one of the main reasons for that, or how I tend to use it for learning, is that you can see an extraordinary number of, say, a group of for example, plants that you're interested in. So I really love the genus Brodia. It's this plant genus. It's a plant that blooms usually in the summer, loses its leaves before blooming. And it's restricted to the west coast of North America. And there are over 20 species. We only have a few in Oregon. Most of them are concentrated in California. I would have to visit California for years and years and travel the whole state to see the amount of species and the amount of observations I could see in a few hours going through iNaturalist. And so I have taken that opportunity to learn the genus, and it provides a pretty amazing way to see a lot of specimens of things you're interested in a very short time period. So it's really neat in that sense. It is good for research, but it's also imperfect for research for all of the reasons that also make it an interesting platform. And that's that it is crowdsourced and anybody can identify things. And so you do get wrong identification sometimes. And that's part of it. That happens. But it is good to understand that a research grade observation, which is when two people agree on what the species is, those get uploaded to a database that is used for research globally. But that doesn't necessarily mean that those identifications are right. They do change. They do get called out. So it's not perfect, but it's really nice. I think I'll jump in quickly and show you what iNaturalist looks like. Let me check. All right, so I'm gonna stop my share here. And then we're gonna reshare desktop. Okay, so I'm gonna try to go through this basically from the start. So this is what, hold on one second. I have to move my Zoom window out of the way a little bit. So when you log into iNaturalist, and creating an account is super easy. So if you haven't created an account yet, you can just go ahead and do that. All you need is an email and a password, basically. It's very quick, very easy. And this is what it looks like when you create an account. This is kind of your homepage. And what your homepage does is it shows you updates on either your own observations or on observations that you have contributed identifications to. So this is my dashboard right now. Clearly what's happening is someone is going through and adding identifications to all of these Pacific sideband snails, which is our largest terrestrial snail in the Pacific Northwest. And although I don't remember doing it, I apparently have added identifications to all of those too at some point. So that's why I'm tagged in there. So basically it's updates on what you've interacted with.
Do I
So
go
that's,
to August?
yeah. Yeah.
Do we go to the App Store to get this, or how do we do it on our phones?
Yeah. So on your phone, you do go to the App Store. So what I was showing, I'll pull that up really quickly here. What I was showing here in the PowerPoint, this is what it looks like on the phone. So it's pretty similar. This is showing only my observations, though. That's what it looks like in your dashboard on the phone. And what you can do here is basically the same as what you can do on the website, but it's a little pared down. So you can, if you see this observe, this camera button down at the bottom, you can observe straight into iNaturalist through your phone camera. And I think that's best to show in person at the training. So I'll do that tomorrow.
Great.
But I am going to show now how to do it through the computer, which is how I prefer to do it. It gives a little bit more options and precision, but both work great. Oops. All right, so back to the iNaturalist website. So that's what your dashboard looks like. But mainly, the really important stuff is this upload button right here in the upper right-hand corner. So that is how you upload photo observations. So you click on that, and you can either drag and drop photos, or you can choose files. I'm going to go to choose files. And so I have these 2 pictures of a lichen that I took today. I'm going to select them both. And then what iNaturalist does when you have multiple photos is it creates multiple observations, and you don't want that. You only want one observation. This is one lichen, right? If this was, if I uploaded 20 pictures here and it was 2 pictures of 10 different lichens, then that's different. We'd be combining them in a different way. But right now what I'm going to do, since these are the same lichen and I want these photos together as part of one observation, I'm just going to drag that on top of the other. And so that's an easy way to do that. Then you have the 2 photos. There's another way to do that, which I'll show in just a second, that is also easy. So the important thing to check here, so I have 2 photos of the same lichen, and we'll talk about this in the in-person trainings too, but it's really important to have more than one photo. Usually you want to make sure that you get the top side and then the underside. Now, some lichens don't really have an underside. They are essentially the same on both sides. And those are usually the ones that are kind of pendulous and hang down. A lot of the more leafy ones have a top and bottom that is distinct and looks very different. And usually a photo of the underside is going to be necessary for identification. So at least 2 photos is going to be good. So I've got 2 here. And what I did here is I took photos that were just at the margin of identifiable to kind of show you that you don't have to have a perfect photo, but you have to have just enough information that I or someone else can kind of zoom in and see what's necessary to see. So I'll get to that in a second. But what I want to check here and make sure, don't worry too much about the date and time. I don't think this time was quite right. Maybe it was. I want to check the location though. So what I'm going to do is I'm going to click on the location button, and that pops up this screen. And this is important. So this shows where the photo was taken. And this is using my cell phone camera's GPS. And if you're like me, you think it's weird that apps are tracking your location. And so maybe you've turned your location off. But what you should be able to do is to choose to allow certain apps to record your location. So you can specifically allow the iNaturalist app to record your location, or you can allow your camera to record your location. So that's what I've done here. And this shows where I recorded this picture, but it gives a really wide berth. And that's because, of course, this GPS is not super precise. Your phone's GPS isn't that great, but it's good enough. And what I like to do, if I know the area really well, and you don't have to do this, but if I know the area really well, I like to reduce the range to make it a little more accurate, because I know that I saw it right within that area. That's not super important. You can do that, but what is important is to make sure, whoops, and I moved it. What is important is to make sure that it doesn't do this, which is have too big of a range that part of it falls outside of the boundary of the Howard Buford Recreation Area. And the reason for that, because partly we want to know, the actual location of your observation so that it could be checked again in the future. That is, that's part of the idea. But the other reason for that is the project, the iNaturalist project, records all lichen observations that are within the Howard Buford Recreation Area. But if there's a little bit outside of it, how it reads that is there's a chance of it being outside of the area, and so it doesn't count that observation. So you just want to double check that. And you don't have to do that in this step. You can do that in multiple steps. But what I'll do is I'll update the observation. Down here, you can add notes. You can say whatever. You can say crappy photos, sorry. But what's fun now is you can get the species name, the suggestion. And I'm really surprised with this one, but even with these fairly poor photos, it gets it right. So it knows, and that's one of the interesting things about iNaturalist too, is part of what is happening with these observations is it's training the computer vision, which is essentially an artificial intelligence. Usually I'm also a little wary of that, but since this is a nonprofit and it's merely identifying organisms, it actually seems fairly neat. Although it will eventually take my job one day, I guess. Hopefully I'll be retired by then if retirement exists. But again, pretty cool. It got it right. What I would say is what I always like to do, also just to see how good it is to check. If you have an idea of what the lichen is and you disagree with it, trust yourself. It doesn't matter. I'm coming around and checking too. Don't trust the computer if it says it's something that's different than what you think it is. So what you would do instead is just type in the name. And so in this case, I'm going to type in what it's suggested, except I'm going to miss a letter. And so that's basically, whoops. That's basically it. Oh, it's acting really weird because I'm in Zoom. Something weird's going on. Okay. So I've got that now, and then I can submit, and the submit button is in the upper right-hand corner. And so that's going to save the observation. And then that's primarily it. But what you can do if you don't want to do that editing in there, or if you want to add another photo, is just click on the observation once it's uploaded. This button right here allows you to add another photo. So you can add plenty. You can add up to 5. Don't add more than 5. There's no reason for it. Usually 2 is going to be enough. But you'll also want to double check that it has gone into this Mount Pisgah Lichen inventory project. And again, if the range of the observation falls within the Howard Buford Recreation Area, then it should show up in the project, unless you identify it as something other than a lichen. That'll really mess with it.
August,
yeah.
question on adjusting that range. Is that obvious when we're on our phone, how that would be, for example, if it was broadening, how we would in fact bring it into a more confined area?
That's a good question. I don't remember on a phone. So let's look at that. If you can remember to bring that up, we'll look at that
Okay,
in person.
thank you.
But you can, once you're here, you can edit the observation. There's always an edit button. You can add more photos. You can add another comment. And then what someone will do is they'll come in and they'll suggest an identification. And this is my observation, so I'm not going to suggest another identification.
So just logistics on this, we could do our hike, do this on the phone, and then we
Yep.
could work with our computers at a desk and then make the adjustments at that point.
Yep.
Thank
You
you.
can do that. Or what I do is I just do it all at home. So I don't use the app frequently. I do sometimes, but
So you
usually
take your
I'll just
computer with you?
no, I'll take the pictures on my phone without using the app and then put them on my computer and upload them. So that's one way you can do it. I actually, most of the time, I prefer to use my digital SLR camera and take photos. And you can do that. You can use any camera you like. The only problem is you have to remember your camera either has to record the GPS location or you have to remember. And the best way to remember is to also take a picture with your camera with a GPS. So if you want to use pictures from your other camera, also take a picture with your cell phone camera to get the GPS coordinates and upload that too. That would be my recommendation. And so you can do either. The great thing about cell phone cameras is they are good enough to get the data that we need most of the time. They'll take clear enough photos. But I like to use my other camera a lot of the time anyway, and a lot of other people do. So it's all up to you. It's a personal choice. The important thing is just getting enough information. And so I'll zoom into this picture and you can see, and this is what I'll do looking at observations is I'll zoom in and see if I can see the defining characteristics. And so with this, I can see the defining characteristics of this lichen species. It has powdery cerealia on the margins, these little tufts of powdery granules, which are how it reproduces. And then underneath, on the bottom of it, it has these forked rhizines. And that combination is diagnostic for the species, the green loop lichen, Hypotrichina sinuosa. And so I can just see enough of that to be able to make that identification, even though it's a little blurry, it's not perfect. But cell phone cameras are going to be good enough for our needs. And then the one other thing that you can do, and again, this is not a requirement, but if you are confident of the tree ID, is there are these cool observation fields you can use. And there are observation fields that people have created for all kinds of things. Like this is one that I use for bumblebees a lot. So it has, if you can't identify a bumblebee to species because they mimic each other a bunch, but you can identify it to one of two, this gives you the opportunity to do that. So that's neat. But for us, what I would do is tree species. And with this was on, this was actually on a shrub. This was on a filbert. So, I'm just going to leave it. I'm not going to put it in, but basically you can choose. The reason I'm not putting it in is I think we have a number of filberts out there that are hybrids, and I'm not sure it'll allow me. Oh yeah, I can just genus Corylus. So there we go. So hazel. And so that's how you do that. And that just adds that extra little bit of information. And that is, that's mostly all you need to know here. So you'll get, when someone comes and updates or comments on your observation, you'll get that feedback here that shows up kind of in your comments area here, or you can also see it in your dashboard. And then you can do whatever you want with your profile. You can update, you can put a picture in there. You don't have to do any of that. But that's basically that. And then I'll quickly show the project. If I can move this zoom bar out of the way. So this is what the project looks like right now. And there are 58 observations currently, and that's mainly because I got this guy here, who's great, Jeff, to join the project, even though he won't be an official member of the project, because he'll be moving soon. He is out at Mount Pisgah a lot and is into lichens, so he's added a bunch of observations already. And so this is what it looks like basically, and it'll track who has the most observations. So if you're super competitive, you can you can make it competitive. The most species, all of that. And what's great is you can view all of the observations in the project. So you can look at what other people are seeing. And it's neat. It's a really nice collection of data. And so this is really kind of the heart of what people will be doing. Research grade, again, I'll talk about that quickly. That means that 2 or more people have agreed with the identification, and that means that it then gets ported often to another website, usually GBIF, which is a biodiversity database that is commonly used in research. And so some of these are not going to end up being research grade, and that's because they don't necessarily have enough data to make them research grade. Maybe the picture isn't quite clear enough. So don't worry too much about that. Or, for example, with this one, Usneas, except for a couple species, are really difficult to identify. They require chemical analysis and microscopy. So we're going to mostly avoid Usneas. But if you're curious about what a lichen is and want to see if anyone knows and record it too, that's one that good chance it's not going to get positively identified beyond the genus. So that pretty well gives an overview of iNaturalist. Any questions? Oh, quickly, sorry. Really quickly, I'll just show. You can also see these. You can see the observations as a grid or as a map. So you can see where people are seeing things. You can see the areas that are less, less explored, which might mean you want to spend more time there. Or you might want to check out an area that has a lot of really cool stuff. Like this looks pretty promising. And that's an area I actually marked on that map. I think this has some neat lichens through here. And then really quickly, one more thing that you can do here is you can just see what has been seen in the whole area too. So I'm still in the project. My bad, I'll back out. So just going to pull out lichens, and we're going to pull out Oregon. That went to Oregon City. That's not what I wanted. Oregon as a state. So this is then all the observations of lichens in the state of Oregon. And so you can zoom in, you can explore this. You can go just to Mount Pisgah and take a peek around. I think when I checked the other day, and you can redo the search in a map to mainly just include this area here. So there are about 350 observations of lichens on Mount Pisgah right now. So we will easily double that, right? That's it sounds like a lot, but it's not a lot. So with 20 to 30 volunteers participating, if each of you made only 10 observations, which you'll probably make quite a few more than that, we'd have another 300. So we'll easily kind of double what's been seen on Mount Pisgah. But this is just a way to kind of explore, take a look around, look at what people see in a specific area. And one of the things that's awesome about iNaturalist too is you can just if you're traveling somewhere, you can see what people have seen in an area. I think that makes it pretty fun too. I used it recently for that purpose. So it really helps you identify things kind of based on location too. Got a couple things in chat. Yeah, it is easiest to use iNaturalist through the app on your phone, because it automatically records the GPS of the location, and then you can go in and fiddle with it if you need to. A time frame for this project is starting anytime now, especially after the trainings tomorrow and Saturday, and then going through March, basically, or at least through the middle of March. So I'll probably put kind of a limit more or less as March 15th. And the reason for that partly is that the lichens really thrive in the rain. I find them to be easier to see and identify in the rain during the rainy season when they're moist, because when they dry out, and honestly, with our warm, windy day today, they were already starting to dry out. So I really like to see them right after rain. They change color a bit and texture a bit. They become a little less noticeable, less easy to pick apart from one another. So I like to see them when they're, right after a rain or during a rain. So in my opinion, it's easiest in the rainy season. So that's kind of the timeline for this. That gives people flexibility to, if you're traveling to not worry about missing a couple weeks or a month even. If you're working, to give you, several weekends to spend some time. But how much time you put into it is up to you. I imagine there are a number of you that are just gonna get kind of addicted to finding out what things are. I swear you'll never look at a forest the same way again. Yeah. Any other questions?
Yeah, I have one.
Yeah.
How are we going to avoid duplicating each other's efforts? Are we not going to worry about that? I mean, to identify the same thing several times, maybe?
Right. I would say don't worry about it. I think you're not likely to duplicate one another's efforts too much, because people are going to find different things at different times. If you're out with, if you're doing this with someone else, at the same time, if you're, kind of taking a walk together with someone, what I would say is don't each record the same thing. So you can look for things together, but only one of you record at one time. So maybe alternate what you're recording. So we don't want to intentionally duplicate the lichen observations too much. But what's interesting about the lichens that we're looking for is that they tend to cluster together. So they tend to really be specific to microclimates. So when you find one, it's often an indication that you're going to find others. So, we'll talk about that more in the training, but that's a thing to look for. Is once you find one, look around for more. Yeah.
Kind of a related question. Are you going to assign certain areas to people at all?
No. I'm thinking folks will just kind of go out on their own, figure that out themselves. If you would like some extra, just like, hey, where would you like me to go? Then I can give an idea. But otherwise, I'd say just do what you want to do. And we'll see how that works. If we get into February and it's like, okay, this area hasn't been covered at all, nobody's gone there yet, take a look at this, then I may, just send an email saying, hey, does someone want to go check this out? But I think there will be decent coverage anyway, with folks just roaming around. Any other questions? Great question so far. And you're also welcome, obviously, to hold your questions for the trainings, the in-person trainings.
You've been mainly showing the web, using the website
Yeah.
application. I've been out playing around with just the iPhone.
Yeah.
And so tomorrow or Saturday, you're going to kind of go through using the iPhone, because it seems like
Definitely.
you can upload the image, you can upload the observations, and then it seems like then maybe if you upload it there, then you can go and edit them in the website, or not the
That's
website,
right.
but the desktop.
Yeah. Yeah. It's really
That one.
easy to upload on your phone and then edit afterwards.
Okay.
If you even need to. A lot of the time you're not going to need to, unless sometimes and I've noticed this more with the iNaturalist app than if I take photos with my phone, but there's a good chance you've noticed that the location is off sometimes by a couple miles. That does
Indeed.
happen from time to time, and then you need to pull it back in, and that's just because the GPS in our phones is not the same quality of a GPS in like a handheld dedicated GPS unit, but that's fairly rare.